hello everyone, and welcome to this webinar on Flash and Prep HPLC. Thank you for taking time out on a Saturday and joining us. And it's a beautiful Saturday morning, and I hope to make it more entertaining for you with this presentation. So moving on with the presentation. So the agenda for my meeting is basically I'll talk very briefly about the basics of chromatography and then explain what's the difference between open column chromatography and flash chromatography, and also explain the difference between what we generally term as preparative chromatography versus analytical chromatography, and then move on to explain the actual instrumentation of this instrument, various detection methods available with them, how they are used in pharma industry and in natural product purification. Also talk some applications about it, and finally give you some quick tips and tricks to improve your resolution in general for any chromatography. So yeah, let's move right into it. As most of you might be aware, back in 1903, Mikhail Sweat was the first person to try column chromatography in a gravimetric way, and he separated chlorophyll molecules using open column chromatography. I know chlorophyll A, B, carotene, and xanthophyll were separated using this technique. And since then, a lot of advances have happened in chromatography, and but the principle never really changed. The column still had what we call a stationary phase, and there was a mobile phase that was introduced, or which is in liquid state, which is introduced from top. And sample is dissolved in your mobile phase, and then sample interacts with both mobile phase and stationary phase, and it gets partitioned between both and gets separated in the column. And once separated, they get eluted one at a time at the end. So that's the basic principle of chromatography, which holds true for any kind of chromatography. You name it liquid chromatography, LC, or you call GC, or you look at affinity chromatography or size exclusion chromatography. I mean, there are many, many names to give, but the principle remains more or less same. There is a stationary phase, there's a mobile phase, there's sample. The sample interacts with some, mobile phase and stationary phase. It gets separated based on its properties, and then you elute one separated molecule after another and collect them or analyze them, whatever you do. So the theory doesn't change more or less between different chromatography techniques, but yes, the instrumentation part, or the sophistication of doing the thing has come a long way since Sweat's time. To give you a parallel, there is still an exists a column instead of a gravimetric glass column. Now there are cartridges and metal columns here that contain your stationary phase. You have a mobile phase reservoir and an automated pump that pulls the solvent out from the reservoir and sends it directly into the column. So there is no human intervention required. Then there are injection loops, which can be automated or manual as you prefer, and which introduces your sample. And if you're doing open column chromatography, you are reliant on your eyes. You can only do colored compounds, more or less. Unless you see them, you cannot separate them. So that to remove that drawback, we now use detectors, which would detect for us that okay, there's a new compound coming in, and every time a new compound comes in, this automated valve or fraction collection valve will switch test tubes or send it to a different vial every time a new compound comes, and that's how you separate them. And whatever is not compound is just coming, solvent is coming, it can go directly to waste, and it also gets collected for recycling further. So the process has automated a lot since 1903. Now in almost 100 years, over 100 years, but the theory and principle doesn't change much. And when we talk about open column versus flash chromatography, the reason I still have this slide on is because even today, after over 100 years, people still do open column chromatography to do their separation, and nothing wrong with it, except it's there are some cons to it that we can really move out of. It's high time we switch from open column to an automated system everywhere. So to give you an idea of the differences between open column versus flash chromatography, if you think of it, you know, it takes roughly 34 minutes for a simple two molecule compound to be separated on an open gravimetric column, whereas it would be done in less than 15 minutes in a flash system because a pump is pushing the solvent down. Flow rates are not controlled in an open column, but you can control them in a flash chromatography. There is no question of creating a gradient, whereas you can easily create a gradient in a flash chromatography. And I'll give you more details about what is gradient, what is the use of gradient flow rate and importance of all that as we move in the talk. But my idea is it is not reproducible when it comes to open column, whereas this is highly reproducible. So if you look at this image on the right, the column also, you can very clearly see the yellow and orange bands are not well separated. They are almost coming together down the column. And as you see in the test tube collection also, like this middle test tubes, this one's is a mixture of both the compounds. So you only get first 3 test tubes here and 2 test tubes here, which are pure of compound A and compound B, and the rest of the middle 3 test tubes are not pure. Whereas if you look at the flash chromatography, the yellow band is clearly below and the red band is clearly above. There's a clean white stationary phase gap in between. And that is also seen in your test tubes here, where first 3, 4 test tubes are yellow, then there are 2 white clean mobile phase, and then again orange has started, and then 4 test tubes are full of orange compound. So it's a very clean separation. There is no overlapping and all those things. And that is because you have a very controlled flow rate, you have a good gradient, and it is extremely reproducible. You can do it over and over again and you get the exact same results every time. It's reproducible and repeatable, and separation is, as I showed, is really good with respect to flash chromatography. So in terms of pros and cons, there's one huge pro to open column: it's cheap. There is no instrumentation, there is no special training required, and it's a low pressure system, so there is less risk of things going bad, or high pressure situations that can cause damage. Those things don't happen in open column. So those who can't afford and still want to do separation based on purify their compound based on chromatography, open column is a good option. However, although it may seem cheap, but over the long term, and if you see in a long period of time, over 5 years, 10 years, it is not cheap. It's not even close, because you spend twice or 4 times as much solvent in an open column compared to a flash instrument. Your quality of separation is not good, so you have to do it over and over again to get really good purity. Your solvent consumption is high. You're not flexible. A person is always stuck in front of the instrument. You cannot do anything else if you are doing open column, and it's not reproducible at all. And you always have to be in a hood to be safe. So there are many cons of doing open column in long term. And if you switch to a flash instrument, you get, first of all, you get fast quick resolutions, you get quick separation, you get good flow rates, you get good automation. The user is free. Once he has injected the sample, he's free to go do his work, and the instrument will do the separation for him. We'll talk more about this in future talk. But Yeah, the solvent consumption is significantly lower. It's extremely flexible. You can use different detectors. You can use different cartridges. There are lots of options you can choose to separate your compounds with, and it is extremely reproducible. And you don't need to be in fume hood necessarily. So over the long term, flash instruments is like a one-time investment thing, and if you do that, it'll always be a advantageous thing for you to do it. And yes, the con is there is a one-time investment of instrument and maintenance, and then in terms of consumable cost, yes, you will have to keep buying the cartridge or columns as and when you use. But if you are doing C18, then you can use the same column for over 1,000 runs and no issues. So it depends how you do your separation. Now, coming to a quick distinction between preparative chromatography and analytical chromatography. When I say preparative chromatography, what I mean is both flash chromatography and preparative HPLC combined. That's both of them fall under preparative chromatography heading. And analytical chromatography includes all the other techniques like LCMS, GCMS, HPLC, UPLC, all those techniques. So as we know, in a typical difference when I talk about, as I mentioned, you have this column where you have a stationary phase which is not moving and it's going to be there, and you load your sample dissolved in mobile phase from top, and then push the solvent either using a pump or let the gravity do the job. And while the solvent comes down, the sample is interacting between mobile phase and stationary phase and gets partitioned between both and gets separated over time. And you have the ones that strongly interact with your stationary phase would stay on top, and those who have weaker interactions would go down the column. Now at this point, if you stop at point 3, where your goal is just to separate and identify the number of compounds present in your mixture. So if your goal is to quantify and qualify your sample mixture, then what you are doing is analytical chromatography. But if you continue from this step and you actually continue eluting each one of these bands separately by changing the mobile phase concentration and eluting one band at a time and collecting each one of this band in an individual test tubes separately, like I showed in the previous image, then what you are doing is preparative chromatography. So now if your goal is to not only separate but actually collect individual compounds and use them further in down the process, then you are doing preparative chromatography. So I'll repeat: if you are doing, if your aim of separation is to quantify and qualify your analytical mixture, if you want to know how much of something is present in your mixture and how many of something is present in your mixture, then you are doing analytical chromatography. But if you're interested in simply collecting each one of the compounds irrespective of their concentration and irrespective of how many of them, you just want to collect every compound that gets separated in a different test tube, then what you're doing is preparative chromatography. Now here's a good slide that I'm going to spend a few minutes on. So over here on the right we have analytical HPLC, and over here we have flash and prep HPLC. First major difference I would like you to notice is the back pressure at the very last. So every chromatography system, irrespective of what is the aim, or what is the reason of that chromatography, has to have a pressure monitoring system. Because when we do separation, we generate a back pressure. Back pressure is nothing but the pressure felt by the mobile phase in going down the column because of the packing in the column. So think of it this way: take a big column, take a big tube or a column, fill it up with huge rocks, like big rocks, and fill it up, and then pour water from top. The water would just slide right down, no resistance, and it will just come smoothly down without any problem. Now take the same column, same diameter column, and fill it up with small pebbles and small tiny pebbles together, and pack it tightly. Now try to pour the water from top. Now you will see the water takes a lot longer to come down, and it comes really slowly down, because there is a resistance offered by those tightly packed pebbles. And then finally take the same diameter column and now fill it up with extremely fine sand and dirt, and pack it really tightly. Now pour water on top. It will take really long for that water to come down, because now the sand is offering a huge resistance for that mobile phase to come down. And this resistance offered by the stationary phase or the packing of your column is measured by pressure, in terms of pressure, and we call it back pressure. Okay. So as you can think of it from my example, the bigger the particles that you use to fill up your column with, the smaller the resistance will be and lesser would be your back pressure. And the smaller the particle that you use, higher would be the back pressure. Okay, and this back pressure goes up exponentially. So if you look at now the particle size, think flash as big rocks, think prep as a pebble, and analytical HPLC as sand. Now, the advantage you have with analytical HPLC is because it's going to offer so much resistance. It also means there is greater amount of interaction between your sample and stationary phase, because it takes longer to come down, right? Better interaction results in better resolution. Better resolution results in better separation. Higher quality separation. Higher quality separation leads to better quantification and qualification. But the problem is you cannot load too much sample. Then the sample amount that you load also affects the back pressure. So in analytical HPLC, you are limited to your loading capacity. You cannot go more than few mgs in an analytical system. But that is where flash comes into picture. So if you want to do bulk separation, if you want to do quick separation and want to just find out what is in my mixture, then flash is the way to go about. So flash can load as much as, I'm saying 3 kilo, but it can be even higher. People do flash chromatography on industrial scale also, where 4 5 kg of samples are also loaded at a time. on a 25-50 kilo cartridges. So yes, it is very much possible to do high volume separation at the same time. And on the other hand, prep HPLC is in right in middle of this too. You do not want to compromise too much on your resolution, yet you want to do slightly higher concentration of separation than analytical separation. That's where prep HPLC comes into picture. So this is the real distinction as to where what you use. So again, because you are loading such a small quantity of sample in analytical HPLC, it doesn't make much sense to collect it and use it further. If you're only injecting mgs and then individual compound will be in micrograms, and if you collect micrograms, what are you going to do with that? It's just not really useful much. So other than that, as I mentioned, flow rates as well, because the packing is so tight and the particle size is so small, you cannot have higher flow rates. You've got to be less than 10. I'm saying 10. 10 is too much, actually. It's usually less than 5 ml per minute also. Most of the time, most analytical columns would be working on less than 5 ml per minute flow rates. So the run lengths are really long. It takes really, really long for analytical HPLC to do a good separation. On the other hand, prep is much faster than analytical, but it's not as good of a separation, but it is much better than flash. And flash is super fast. It is truly flash, and that's why it's called a flash chromatography. And it happens. So what would take around 3 hours on analytical HPLC might finish in 30 minutes in flash. And if it's a compound mixture of 10 compounds, flash would separate at least 6 or 7 of them easily, depending on how well you have prepared your method, ingredient, and all that. So yes, flash is not the best technique to do your separations perfectly, but it is a very good technique to start off with. So the pressure limits that each system is capable of handling is also different. In terms of flash, you will only find systems which are 50 bar or less. In fact, Brookie is the only one who gives a 50 bar system. 50 bar is roughly 725 psi. So the Buki is the only one who gives 725 psi resistant systems. Others only give you 20 or max 30 bar, nothing more than that. So I mean, at 20-30 bar, you cannot use particles less than 15 micron. Even 15 is too much. You cannot go at 20 bar system. You can hardly go up to 30 micron particles. So like I said, smaller the particle, better the separation. So that is where your resolution is compromised. But the advantage is very high loading capacities. On the other hand, prep HPLC can go up to 300 bars also. That's about 4,000 psis. And yeah, with that much back pressure limit, you can really do high quality separation. So to continue with that example, I would say all 10 compounds would be separated on prep HPLC also, but the resolution might not be as good compared to analytical HPLC. But yes, you will be able to separate them over the time, and you can do larger quantity also. So, and in terms of how the columns look like, the below picture shows you what the column looks like. So in terms of flash, they are actually, we call them cartridges, and they are packed in a polypropylene material. And because the pressure is not so high, polypropylene is good enough to resist those pressures. And you can get this cartridges from 4 grams to 5 kilo to 50 kilo as well in an industrial scale. And then coming to prep HPLC, because the pressures are going to be high, particle size are going to be smaller. We need to switch from plastic to metal because plastic cannot handle so much pressure. So we have metal columns, but these are really big diameter columns. And eventually, in terms of analytical chemistry, it is really small particle sizes and really small columns as well. So I hope this helps you understand the difference between prep chromatography and analytical chromatography as such, and why and when you should use what. This is what prep and flash HPLC instrument looks like. This is Buki's one of the Buki's flash and prep combined instrument. As you can see, it is a very compact instrument, and there isn't much moving parts in it. This 4 valve thing, 4 dots, was where the solvent lines would go in, and up in the front you have your touchscreen where you control your instrument with. At the bottom over here, this is your pump. This is where you load your column or cartridge, and this is the test tubes where whatever is getting separated goes here. These are the detectors over here. internally the detector will do the job, and then the separated compounds will come out over here and start getting collected in individual test tubes based on what the chromatogram comes. Right. So this may look slightly different from what you are used to of seeing in terms of analytical HPLC systems, but the parts and the idea or the theory is exactly same. In analytical HPLC system, you have different sections. One section will have pump, one section will have your injections, one section will have your detection system. So it's the same idea, but this is I would say a better design, a sleek design. And coming to like the real internal design, if you want to think of it that way, it looks something like this. This is one of our just UV only system. So the way it will look like is it starts from here where you have your solvent lines. We have NRV valves over here, and this is your pump. The pump sucks the solvent from the bottles depending on what gradient you created in your software, and the solvent moves along this path. This is our distribution block where it actually creates the gradient, mixes the solvent as you mentioned in your software, and then it continues over down to your column. It enters your cartridge or column over here. There, and this port over here is your injection valve. Over here, you inject your sample. Sample injection happens here. And then as you inject your sample, your mobile phase takes the sample with it and comes inside the column. Your separation occurs in the column, and then one individual or multiple individually eluted compounds will follow along, come to the UV detector, and you will see your detection response in terms of chromatogram in your screen. And then they go into this fraction collection valve, which either would send the separated compound into your fraction collection test tubes or to waste, depending on what is coming. So if nothing is coming, just mobile phase is coming, then it will go to waste. And if some compound is coming, it will go to fraction collection. So it's pretty much a very simple design as such. And depending on what detector you use, things might change here accordingly. If you have ELSD detector with it, then you will have a bifurcated, you'll have a T valve here. Some goes to ELSD, some goes to UV, and then both come over here or whatever. So based on what detection systems you use, some slight design changes might happen. But overall, this is the design of any preparative HPLC or even to some extent analytical HPLC system. Now, like I was mentioning, the main difference that happened over the years between open column and a flash system is the pumps, introduction of pumps. Think of pump as the heart of a chromatography system. It really defines how the performance of your separation of your system is going to be. And there are two kinds of pump that Buki offers. And this is the most standard pump that you guys are used to of seeing. These are the HPLC, standard HPLC metal pumps that can resist pressure of 300 bar or more, 200 to 500, 600 bar pressure resistance capacity pumps. And the way this pump works is basically each one of this has a piston in it. And this piston work in parallel. The way they work is one piston is pulling the solvent from the reservoir bottle, while the other piston is pushing the solvent in your column or cartridge. And while you are running constantly, there is this push pull going on in the pump. But the one advantage that, or I guess disadvantage you have for this pump is during this push and pull that goes on, there will be a time in moment where nothing is being pulled or nothing is being pushed in the column, and that creates a dead volume. And that dead volume enters your column, and it causes all the issues with your separation. It causes peak broadening, it causes your peak fronting, tailing issues, and all those things. Peak overlaps happen sometimes if they are close line compounds. So to avoid this this issue, Buki came up with this design of this unique 3-piston pump design. So where there are 3 pistons in this pump. This is 1, 2, and 3 pistons radially arranged in one configuration. So the way this will work is same idea. There are 2 pistons which are pushing and pulling, but the time when there is nothing being pushed and pulled, there is this 3rd piston in reserve which is ready to push solvent in column when nothing is being pushed. So they keep working in circular motion one after another. So what we produce is a pulsation free flow. When you have a dead volume going in the column, what you create is a pulsed flow. And to negotiate this pulsation in your prep pumps or prep or analytical pumps. What many manufacturers use is what we call pulse dampeners, which is nothing but a third piston or a syringe pump that pushes that solvent in. But that is an additional cost, and there's a need for synchronization and all those things. But with this three piston pump, you solve that issue completely. We automatically produce a pulsation free flow, and the pump is also very resistant, and it can resist up to 50 bars of pressure. Like I said, is unique to us, and it helps. The advantage you get with 50 bar is you can use smaller size particles in your cartridges and get better separation. So we have customers who have been able to use even 10 micron or 10 micron particle size in the flash system. And if you remember my table from previous slide, I said 5 to 15 is actually in prep region. So with this kind of pump, you can even do prep type separation in a flash system. Now coming to detectors, think of detector as your brain of your flash system. It is what it is the one who identifies everything that is eluting. And there are many choices. You have many choices, and most common and most commonly found detector is the UV detector, or sometimes UV vis detector, depending on the sample. And it is easy to use, easy to manufacture, easy to maintain. So that's why it's so common. The main advantage is you can detect almost any chromophoric compounds, anything with a benzene ring, anything with a resonance, anything with good chiral chemistry in it can be easily detected using UV detector. And it has good sensitivity for chromophores, no questions. It has very good sensitivity. It is gradient compatible. It is non-destructive. So that is one of the good reasons why UV and UV vis detectors are so common. Other than that, another more commonly found in flash systems and prep systems is ELSD, also called as evaporative light scattering detector. This detector is more of a universal detector. It detects everything except volatile compounds. So if your compound is more volatile than your mobile phase, then you cannot detect it. Otherwise, it detects everything else. It's also to some extent a mass-based detector. I will explain you why in a minute. It has a medium to high sensitivity. Yes, it is gradient compatible. But unfortunately, it is a destructive technique. What goes in ELSD is gone. You cannot collect it, and it is very easy to use, no question. And the third most common detector is mass spec detector. Any compound that you can ionize can be detected using mass spec. Also, medium to high sensitivity. Gradient compatible, yes, again, very destructive. Whatever goes in is gone, and not easy to use. I have worked on mass spec, and I know the qualification and quantification using mass spec is not easy. It needs some training, it needs some experience, it needs time to learn mass spec. Back in the days before ELSD and mass spec took over, there were RI detectors, refractive index detectors, that were used. Again, it's a universal detector, can detect almost anything, but it doesn't have a very good sensitivity. And the biggest disadvantage is it is not gradient compatible. You cannot use gradients. So it was, this was RI was the time when it was being switched from open column to an instrument. The way the transition happened was, first only people used pumps to push through open column, and then they started using this RI detector to detect compounds, because those were not colored, and they wanted to measure those UV compounds, but were not able to detect them. So RI was used instead. And in those days, it was considered a very good detector and a very universal detector. But nowadays, it's not as so common. In fact, I would say it's almost extinct. And then there are special detectors like fluorescence detectors. Yes, it is very specific detector for specific compounds that fluoresce. And for fluorescing compounds, it's extremely sensitive, and it gives very good results if your compounds are fluorescent. So yes, it is gradient compatible, and no, it's not disruptive technique. You can collect it, everything that goes in there. Again, not easy to use. You need training. You need to understand how it works, and user training and everything is required. But overall, I would say UV, ELSD, and mass spec are the three most common detectors you will see in flash and prep instruments. So let's talk a little about UV while we are on the subject. I mean, I'm sure all of you know all this, that you have You have UV light that enters your sample. Your sample absorbs some of those UV light, and the difference in absorption from your I over I naught will give you your absorbance. And in terms of Beer-Lambert's law, it's molar absorptivity times concentration times your path length. And playing around with different any one of these parameters, either your concentration or path length, you can adjust the absorbance of your sample. And most manufacturers will offer you different path length cells, flow cells, to use with your science instrument. So if you have a very low UV absorbing compounds, then you can opt for a bigger path length instrument, and that would increase your absorbance significantly, and you will start seeing the peaks that you were not able to see before. The 0.3 mm one, this one, 0.1 to 0.4, so the standard size is 0.3 is the most common one actually. But like I said, depending on your requirement, you can ask your manufacturer to provide you with a better path length cell to have a better sensitivity. With advents of diode array detectors, or what are also known as photodiode detectors, it is possible to do multiple wavelengths at a time. So you can scan the entire 200 to 800 nanometer UV vis range at a time, and get information of the entire spectrum in single run. So it's one of the major advantages of DAD. And with combining the intensity versus time versus wavelength plots, you can even find the lambda max of your unknown compounds. In a typical flash instrument, even today, after using instruments, people tend to check the TLC of every eluted fraction. That means they will take TLC of each test tube to see if the compound is still pure or not. Although chromatogram may show only as one peak, but there might be multiple compounds that might have eluted under a same peak. And to avoid this issue, they do TLC of each compound. But when you use DAD detector and you plot like this 3D plots, you can clearly see, like for example in this one, if you had only seen the 2D chromatogram of this, it would have looked like a one big peak. But the 3D peak clearly shows that it's a two compound illusion. There are two compounds in that single peak. So that eliminates the need to do TLC of each test tube to find out that is this peak pure or not, whereas the second peak is quite pure. It's only one compound. So that's one of the major advantages of DAD detectors. And of course, if you have unknown compounds that you are not expecting, you can find the lambda max of those compounds using this DAD detector as well. So next time you can use that wavelength to separate that compound as well. So some of the major advantages, like I mentioned, it's easy to use, extremely reliable, not expensive, gradient compatible, not destructive, and fairly specific in terms of chromophores. Limitation is if it is not a chromophore, you'll lose it. You don't know. You won't find out. There are another major disadvantages. There are solvents that absorb UV light as well. Like for example, if you look at ethyl acetate UV spectrum, it easily absorbs up to 220 nanometer also. So if your compound is also absorbing in 210 to 15 wavelength range, then you are doomed more or less, because both solvent and compound are absorbing, and the peaks will overlap on top of each other. So it will be very difficult to separate. Baseline correction will be very difficult. And it's not quantitative technique. You cannot quantify amounts using UV detector. Coming to ELSD, evaporative light scattering detector. The way that works is, your compounds elute out of your column and come over here. This is the nebulizer or a nebulization step, where a small quantity of your sample gets nebulized with high pressure air coming from here. It breaks down the small droplets into extremely small droplet or aerosol particles, which travel along this tube, drift tube. Now, a standard design, or what I say, a normal ELSD would actually heat this drift tube and help the solvent molecules to completely evaporate out of your sample, and only let the sample reach your detector at the end over here. That is what typically happens in a ELSD. That is why it's called an evaporative technique. It evaporates your solvent off completely. And then the individual sample molecule reaches over here at the end, where a laser is firing in a parallel direction And each time a sample molecule passes through laser, it scatters that laser in different direction, and the amount of scattering is directly proportional to your sample concentration. And also, because there's a long drift tube, it is going to get separated based on mass, just like a time of flight mass spectrometer detector. So based on the velocity and the weight of each sample, the lighter ones would reach first and the heavier ones would reach later, something like that. So, but also, it also depends on what is eluting out of your column. So um, if out of your column two compounds are eluting in a single peak, and one of them is lighter and one of them is heavier, then the lighter one would reach first and the heavier one would reach later. Okay, I hope that makes sense. Now, although this is what a standard ELSD does, but Buki has a patented ELSD technique where the amount of sample that we take in for ELSD is so small. It's only 30 microliters per minute. This is our specialized patented splitter that splits the flow of eluted column flow into two, where 99.9% of your sample goes to UV and only 0.1 comes to ELSD. Like I mentioned in my previous slide, ELSD is a destructive technique. So you ultimate goal of any prep or flash HPLC system is to separate and collect all the compounds. So you want maximum yield. So sending a huge quantity to ELSD is not advantageous for anybody. So we have a technique where we only use 30 microliter per minute of your sample, and that is less than 0.1% of your sample. And because the quantity is already so small, when it gets further nebulized, those particles are so tiny that the solvent actually evaporates on its own at room temperature in this drift tube, including water. So even water would just evaporate out while it is traveling through this drift tube. So we do not need to heat anything in our system, and that is the major advantage of a Buki LSD, where without heating you can measure all the compounds. And these are the typical compounds that you would measure using ELSD, and which would be very difficult or nearly impossible to measure using UV. And with mass spec, it will be difficult to isolate or identify each one of them, because their fragmentation pattern won't be so unique every time. So some of them include, essential oils, lipids, your carbohydrates, steroids, surfactants, histidines. These are sugars. These are some commonly used compounds where ELSD has given an edge. So another advantage is it's not solvent dependent. You can use ethyl acetate. You can use any solvent, that is any organic solvent, and we know all organic solvents are going to evaporate easily, so not a problem. Another major advantage is, there will be impurities that you didn't know existed in your compound would easily be detected by ELSD. And it is also a relatively quantitative method. You can kind of identify how much concentration of your sample is present based on the peak height. Here's an example of that. So this was the actual concentration of your sample of the three compound mixture that got separated using ELSD. And using peak height, we could estimate almost pretty accurately that first compound is 40% and last compound is 20% and stuff like that. But now if you compare the same thing with respect to UV, although the concentration is same, but their absorptivity is so different that peak 1 and peak 2 are not really absorbing any UV light, whereas the third compound is absorbing almost all complete UV light, and that peak is huge, whereas peak 1 and peak 2 are so small that you cannot quantify anything using UV. And that is, I think, a big disadvantage of UV. It looks very good. ELSD seems to be a very perfect technique. And there is one major disadvantage of ELSD, is that the mobile phase needs to be evaporated. It needs to be volatile. Up to water, yes, we can volatile. We can evaporate everything, but things like dimethylformamide or DMSO, it's very difficult to use with ELSD. The solvent won't completely go, and then the solvent will become a peak. Also, another disadvantage is volatile compounds. So, for example, vanillin. That's a essential for that makes your vanilla. Um, so that compound is extremely volatile. I think it vaporizes at 45, 50 degrees something. So no matter what solvent I use, the compound is going to get evaporated as well, and nothing will reach the detector. So if your compound is volatile, and if your solvent is not easily evaporated, those are the two times you cannot use ELSD. Otherwise, every other time ELSD is extremely useful. And the modern techniques and the modern instruments are now able to use multiple detectors at the same time, and that's the best thing about it. So for example, this chromatogram is a mixture of three compounds. One of them is non-chromophoric and two are chromophoric. And as you can see, in this case, we used both the detectors at the same time. And the very first peak is for cholesterol acetate, and the UV yellow and orange lights color graph are completely flat here. They didn't detect anything, whereas ELSD detected it. And then moving on, the other two compounds got detected by UV and ELSD as well. You can clearly see there's a green peak behind as well. So ELSD would detect all three, and UV will detect only two of them. So that's the real power of modern instruments, that you can use multiple detectors at the same time. You can do the same thing with UV and mass spec. You can do the same thing with ELSD mass spec. All that combinations are available and possible to do. All right. So now coming to, I've told you a lot of story about how the instrument works, how it's purified. But you will ask me like, where do I use it and why do I use it? So here's where and why. So in a typical organic synthesis lab, you have multiple steps. Step 1, 2, 3, 4, 5. You do all your reactions, and at the end you have some compound that you think you have prepared. But whenever you have your final step done, it won't be like a ready-made solid compound. It'll be dissolved in some kind of solvent. It will come with some non-essential solvent, or some non-essential something that you need to remove, and that we do you at evaporation step. And at this step, one of the Bookie products would be very useful is a rotary evaporator. And rotary evaporator is used to evaporate your solvent, distill out or distill the solvent, or to repurify your solvents and things like that. And I will come to it in a minute and explain how a rotary evaporator works as well. And as Dr. Ramakrishnan mentioned, Bookie is the pioneer in making rotary evaporator. We are the ones who developed this technology, and we know really well how to do rotary evaporator or evaporation techniques. From there, what you would usually do is to some kind of analytical chromatography. You need to first identify how much of your compound is present in that final mixture that you made. We try to make one compound, but we know we have a lot of impurities that are made with it as well. So we need to identify the concentration of impurity. We need to identify the concentration of your actual compound of interest. And that is typically done using either TLCs or analytical HPLC more often than not. And uh, you know TLC is easy. It's quick. It gives a lot of information pretty quickly. At least the question of how much is pretty much, or how many rather, how many is pretty quickly answered using TLC. But a lot of time TLC is not useful because the compounds are non-polar and they don't get separated on a TLC plate. Then in those cases we have to do HPLC. After you have found out, okay, my compound is 80% pure, and now I'm ready to, I'm ready to remove the other 20% and collect this 80% to 100%, convert this 80% to 100%, and move on. That's when you come to preparative chromatography. You can either do only flash, or you can either do only prep, or you can do both flash and prep to achieve the highest purity and resolution that you are looking for. I'll come to it in a minute and explain you how that works. And again, after doing prep HPLC, you will have a lot of mobile phase eluting with your compound of interest, and you need to remove that mobile phase as well. So again, rotary evaporator can be useful for that purpose, but a lot of times your compound now is heat sensitive and you cannot use rotary evaporator. You need a very gentle technique to remove those mobile phase. In that case, a freeze dryer or a lyophilizer comes into picture. Bookie also offers one of this lyophilizer for this purpose only, for freeze drying your sample. And I'll also explain how that works in a minute. And after you have finally, separated your compound, purified your compound, and you have your pure compound in your hand, then two things happen. One, it goes into your analysis department, QC analysis department, where you analyze its NMR, you check its NMR, you check its FTIR, and confirm the compound is what you think it is. And once you are sure that this is what it is, then you go into clinical trials, the test it for its efficacy and all that. And once the clinical trials are over and you're sure that this is the drug I want and this is what it is made for, it goes into production and quality control. where again all the steps are repeated pretty much. All the synthesis steps, all the analytical testing, prep separation as well occurs on a larger scale, much larger scale. And after that, you can use spray dryer or an encapsulator to make the final formulation, either in a powder nasal form or tablets or capsules or whatever it is. So all that happens, and in QC department also, you constantly check the quality of product at each step using melting points and NMR and HPLC and other systems. So this is how typically a drug is made, right from conceptualization of a drug to final product. So the part where I said you can use both or one, this is how it's used. So let's say you did some reaction, some reaction. You have two compounds from A and B. This is a very simple example, but I hope it will make sense. And at this point, you do your analytical HPLC right here. After you have finished the reaction, you take your mixture and put it in analytical instrument, and that's where you will find out that okay, this is 78% and this is 22%. A flash or a prep instrument cannot tell you how much of individual is present. That's not possible. Okay. And then you know that okay, this is how it looks. So you take this, okay, if it is 70% pure, I can start with flash chromatography, do a quick separation and improve the purity further to 80-89%. It does not completely separate both the compounds fully, so you only get your resolution wasn't good basically, so you only get 89% purity. And this is the limit for a flash instrument for this compound. So now you have to switch to prep HPLC, use a smaller size particle in your stationary phase and do a high resolution separation. And that's when you're going to get almost 90% pure of compound, this compound of interest, and you'll remove all this other impurity completely. So this is how you use flash and prep instrument in a pharma workflow, or in almost any workflow actually. Typically, only flash can take you up to 90-95% at best. Beyond that, you have to go to prep chromatography. So how does a rotary evaporator work? Basically, you have a bath, a heating bath, which you heat up to whatever temperature of your solvent is that you're trying to evaporate out of your sample. And this flask lowers down in this water bath, and the flask rotates. The reason it's called rotary evaporator is because the flask is rotating, and by rotating, we are increasing the surface area that is exposed to hot solvent, hot water bath, and the evaporation is quickened a lot compared to a normal distillation unit that we know of in simple glassware design. So your evaporation is quick. The solvent vapors will travel through this vapor duct, and over here we have a condenser which is cooled, chilled at a low temperature, and as the vapors come in contact with this cold chiller, they condense and come back as pure solvent and get collected in this reservoir. So this is a very simple design, but a very effective design to remove your solvent, to distill your solvent, or to repurify your solvents, or to concentrate your sample. There are many advantages of this instrument in that applications that you can use it for. Please use your phone's QR code to take an image of this QR code, or scan this QR code, and it will take you to Buki's rotary evaporator information, and you will get a lot of information about rotary evaporator from there. Coming to freeze dryer now. So like I mentioned, after you have done your separation, a lot of time you have compounds, natural products or biomolecules, or even synthetic products that are heat sensitive and who cannot afford to be placed in a hot water bath of rotary evaporator. The compound will denature or decompose or change its properties. So we cannot afford to heat them. In that case, freeze dryer is your solution. So what happens is basically you take advantage of the phase diagram of your compound. Think of the solvent, not compound, my bad, solvent that you're trying to remove. So let's take an example of water. Let's assume water was your mobile phase. Then freeze drying, first and foremost, freeze drying always happens below triple point of your solvent. So whatever you're going to do, you're going to work below triple point. Okay. And first step in freeze drying is to freeze your sample. You physically freeze your sample below 0 degrees Celsius in case of water. And then once it's frozen, you load it in the lyophilizer in sample vials, or you can use this. This is a rack, but you can also use your flasks. You can use dishes like this ones. or you can just place it right as it is also inside the chamber. There are many different ways you can load sample, but the idea is you load a frozen sample in there, and then using a vacuum pump you reduce the pressure inside the chamber big time, and bring it below the triple point and below the liquid line, and come to the solid, from solid to gas phase directly. You sublimate your ice from solid to gas directly. In this area it is in solid phase, and in this area it is in gas phase, so you go from solid to gas directly. Okay, and then this because the pressure is low over here and high in this condensation zone, the vapors automatically go from low to high vapor pressure area and start condensing again on this condenser, and the pump continuously keeping the pressure low in the system. This way it is a very gentle technique. So the structure of your sample, the shape, the properties does not change at all. Only the solvents gets frozen and removed completely, and this way you get very nicely dried compound. However, I would say it is a slow technique. It takes, sometimes it can take even 24 hours or 48 hours to dry one sample completely, depending on the content of solvent present, but it is very gentle. Your sample is never damaged, and you get very good dried compound. And again, same QR code. Please take a picture or scan it, and you'll get a lot more information about lyophilizer and everything. Just like we talked about organic synthetic products, we have natural products as well that we can use to produce drugs nowadays. We rely a lot on natural products these days to produce biosimilars, biomolecules and things like that. Typically, this is an example of a cannabis drug. Cannabis is very strong in US and Western countries now. The way that works is, you harvest your plant, whatever material you are using in natural product, you extract your compound of interest using some kind of extraction technique, either by dissolving in a solvent and keeping overnight, or using CO2 extraction, supercritical CO2 extraction, or rock select, or some extraction technique. It could be many. There are many ways to do that. After you have extraction, again, the idea is you will have some unwanted solvent with your compound. You can use a rotary evaporator to remove that solvent, and then you have a concentrate of your compound. You can load that on a prep chromatography system and separate each one of those mixture of compounds into individual compounds. In this case, it's CBD, THC, and TCHA. And then take it further again to remove the excess eluted solvent with it in a rotary evaporator, remove that, and then you have your pure compound in a dried form, which you can use further to either make capsules or powder in spray dry forms or whatever you prefer. And from there it goes out to drug. So the workflow is very similar for most products. And now coming to the options you have in terms of modern instruments. This is all that Buki offers. Buki has 3 instruments for flash chromatography, 2 for prep, and 1 for flash and prep combined. This is a combined instrument. So the main difference between these 3 instruments is this 805 is our latest launch. In 2021 only we launched this instrument. This is for especially for our academic customers who have low funding and low budget customers. It has these are for someone who wants to switch from open column to a flash instrument. It has every basic thing of a flash instrument in it. It has 2 solvent lines, it has a UV detector, it has a column cartridge holder, fraction collection area, and it looks almost as same as this one as well. So it is a very unique design with the same 50 bar pump. So you can get a lot of things done using this instrument if you switch from open column to a flash instrument. So it's a budget friendly instrument. And then we have, so this one, like I mentioned, only has a UV detector. And if you come to 810, it is a UV visible detector, and it has many additional safety and software features that you get with this. And then you have 815, which is a UV vis and an ELSD combined detector. Everything is integrated in a single instrument. So you have UV, UV vis, and ELSD in it. Similarly, this is a prep only system. So this 3 system, all of them go up to 50 bar back pressure limit, 50 bar, 725 psi. Whereas this 2, 830 and 835 can go up to 300 bar or 4000 psi back pressure limits. And everything else is similar. This has a UV vis detector, and this has a UV vis and an ELSD detector. Whereas this 850 is the most advanced one and has everything in it. It has a It has a 300 bar capacity instrument. You can limit it to 50 bar if you're working in a flash mode, and you can go up to 300 bar if you're working in a prep mode. You can load a column, you can load a cartridge, you have UV, Viz, ELSD, everything in it. So it's top of the line, everything in it. Again, this QR code will take you to our website and show you more, or give you more details of all these instruments if you're interested in moving on from open column to flash, or if you want to upgrade your flash instrument to ours. Okay, we talked about its application. Let's see where all to today flash instruments are used. I would say, if you have taken a medicine in your life, that medicine has gone through a flash instrument once for sure, 100%. So this is a typical example where an anti-malarial drug was purified using flash instrument. Another example where a lot of times it's not the drug that is the interest, it's the impurity that becomes of interest to the researchers, because they need to understand what is that impurity that is being formed in the reaction, and they need to adjust the reaction so that that impurity is not formed. So isolating impurity is also a very important step in drug discovery. And that is being, and the difficult part with impurity isolation is their concentration is going to be super small compared to your actual compound of interest. So isolating that particular single compound is always difficult, but it can be done using a flash instrument also. And of course, if required, we can switch to prep. So coming to applications, we have biomolecule application as well. You can purify peptides. This is a peptide purification where we use both flash and prep to purify that compound. You can even use it for natural product separation. In this case, it is a clove oil. And this is just normal clove. If you can have essential oils in that clove that you can extract and use it as medicines for Ayurvedic medicines. Good example is Patanjali is using our flash prep instrument to do something similar. They're using for curcumin and clove oil and whatnot. So that's possible. And of course, like I mentioned, you can use the flash and prep combined for many applications. This is again vitamin E isolation from wheat germ oil. Now coming to resolution, like I mentioned, I'll give you some tips on resolution. So what is resolution? Resolution is the ability to separate two peaks completely, right? This is a resolution of 1.5 where it has not completely reached the baseline, whereas this is 2 where it is just touching the baseline, and then it is well separated, then it's a 4 and above. So ideal world, we want every peak to be at resolution 2 at least, but we are happy to work at 1.5 also. So the way you calculate resolution is based on your theoretical plates, your selectivity factor, and your retention factor. So what is retention? Retention is basically how well your compound is retained on your stationary phase, and that is calculated by separating the retention time of the peak minus the TM, and you get your retention factor. And selectivity is how much does your stationary phase select one compound over the other, how well it is separating the two peaks, basically, is selectivity. And efficiency is how fast and how good the separation is, right? So there are different parameters that you can adjust to improve your resolution. Okay. The most impactful and the highly useful parameter to change would be your stationary phase and mobile phase combination to get best separate resolution. So selectivity has the highest impact on your resolution. Then efficiency has it, and efficiency is affected by sample size, particle shape, flow rates, and things like that. And retention is affected based on your mobile phase strength. Looking at selectivity in terms of stationary phase choices, you have many different types of stationary phase. Just like we say, like dissolves like. In affinity chromatography, like adsorbs on like. If you have a very polar compound, a very polar stationary phase would be ideal to separate it. And if you have a very non-polar compound, a very non-polar C18 would be ideal for it. So silica is good for normal phase separation, C18 is good for reverse phase separation. And then if you have a compound with a lot of amine groups on it, then you have a choice of an amino cartridge. If you have compounds like carbohydrates, then diol is a good choice. And alumina is also used in case where silica is not a good job. And alumina helps in doing a pH-based separation also. You can get alumina in neutral, basic, and acidic media as well. So, and these are just a few examples that Buki offers. And, Buki has all these different cartridges in all different shape, size, and quantities, from 4 grams to 5 kilos, and from 5 micron to 60 micron particle size, and all kinds of variation. We have over 180 different cartridges that you can choose from. But here's a quick tip. Typically, like I showed in my workflow, you do TLC to identify how many compounds are present, and then switch to analytical or prep HPLC for the purification. So if your RF values are between zero point 0.2 to zero point 0.5, that means the silica is really retaining it well. So you got to do normal phase separation in your flash chromatography. But if your RF values are in higher than zero point 0.7 and zero point 0.8 and above, then silica is not really retaining them, and that means your compounds are non-polar. And then in that case, reverse phase or C18 separation would be ideal when you do your flash chromatography. And if you have very low, very high, I mean, I'm sorry, if your RF values are lower than 0.1, then it has a very strong interaction with silica, and I would suggest to not use silica or alumina and go for a specific cartridge like amino or diol, where compound functional group is used to separate them. Coming to your selectivity based on mobile phase, this is a Cinder and Kirkland triangle. If most of you must be aware of this, and you have proton donor, proton acceptor, and a dipole ends of the triangle, and these are different groups of solvent that you can use to select ideal solvent for your separation. And I can give that example using this TLC of this compound. This is the compound of this three mixture, and while separating, if you use only chloroform, which is a very proton donating solvent, then these two compounds didn't really separate well. But if you use a very dipole solvent, they do separate, but not so well still. And if you use tetrahydrofuran, which is a hydrogen accepting solvent, then it is doing a good separation. All three compounds are well separated, right, individually. So we conclude tetrahydrofuran would be a good solvent choice in this. And then you can use that solvent in your flash instrument. And in flash, there is a possibility to do gradients, and that's when you can play with solvent combinations. In this case, I added hexane in it, and you have a 1:3 ratio of THF to hexane, and you can clearly see the separation has improved big time. So this is how you optimize your selectivity based on mobile phase. And then in terms of retention, you can make gradients to improve retention. Like, for example, in this case, when it was a clear step gradient, this 6-compound mixture wasn't really well separated. But when I mixed a linear and a step gradient together, they got really well separated. So playing with your gradient will really help you improve your separation also. Now, coming to efficiency, there are many factors that affect efficiency. So let's say when we talk about mobile phase, the flow rate is very critical. Choosing the correct flow rate will improve your separation quality big time. And the nature of your chemical solvent, like I said, like dissolve likes, because eventually you need to elute your compound from the column also. So the polarity strength of your mobile phase also affects your separation. Comes to stationary phase, again, particle size and particle shape that you use in your stationary phase has a huge role in your separation efficiency. Smaller the particle, better the resolution. Spherical the particle, better the resolution. Also, surface area and pore size are secondary factors that would affect your separation as well. Solute factors, that means your sample, how much the quality or the properties of your sample affects also. So the amount of sample that you load has a big impact on the separation quality. So you need to optimize this sample loading also big time. Although the column or cartridge can handle larger sample load, but it is not always advisable to load the max quantity. Sometimes reducing is also required. Other factors as peak shapes that affect, depending on the peak shape, you can understand what is wrong in your chromatography and adjust that particular parameter to improve your efficiency. And some mechanical factors also come into picture, like I mentioned, dead volumes. Because you have a pulsed flow, then you will have a dead volume, which is going to affect your efficiency. So you can improve that by switching to a pulsation free flow and things like that. So my take-home message is very simple. Automated flash and prep HPLC systems are going to have a very good impact on your purification process. You're going to get very high purity samples using an automated system. And if you want to improve your efficiency, if you focus on efficiency, that means if you focus on the particle size, flow rates, and all those things to improve your resolution, it is going to be a quick step. It is going to be a quick result, but it is not going to be as effective. But if you spend time and identify the correct stationary phase, the correct mobile phase ratio, and the correct concentration of mobile phase and all, correct gradient and all that, then it will be a time consuming thing, but you will have a very good separation, a very high resolution. So start with selectivity and then go to efficiency to get a good resolution. And lastly, just take last 2 minutes. I know I took a lot of topics and I covered it in a very quick fashion. And to be honest, I just touched the tip of the iceberg of each one of these topics. But Bookie themselves also have their own course on chromatography, and I will cover all these topics in far more detail in each one of these webinars. I know individually I'll spend an hour on each of this topic and talk a lot more and in more details about this. And if you're interested to learn more, please register over here using this link or this QR code, and you'll be able to attend all these webinars that we'll do over the year. And you can also, if you register, you get a free Chromapedia, which is the complete guide to all this topic in terms of PDF as well. So yeah, you're welcome to register with us as well for more details to learn about these topics.